All right, folks. Hello. Happy Wednesday. Sorry, I'm so used to having this on Tuesday, so Wednesday really throws me off. I'm not sure how I'm supposed to act on a Wednesday. Hi, my name's Jim Gall. I'm the host of the Grit City Think and Drink. Happy to have you here. Normally, this is the second Tuesday of every month. So don't be fooled because it will return to Tuesday in December. So I will give a quick intro to what we're doing tonight. I'll introduce our speaker, and he will give you a talk to knock your socks off. We will then give away some prizes, and then we'll do a Q&A with the speaker. So you can ask him any strand on the human genome and see if he knows it. If not, he's buying beer. He doesn't know that yet. So, okay. The way we do this, so the second Tuesday of every month, we have a different speaker all over the board. So we've had speakers on dance last time. So we're doing human genome this time. We're doing education and place next time. And we're looking for ideas for 2026. So if you have speakers or ideas for people that give talks, definitely let me know. And I'm happy to contact them and see if we can get them scheduled for 2026. So if you want to join the to get messages about the upcoming talks, you can scan the QR code and just add your name. I do not do anything but set out info on the talks, so we don't ask for money. That's all under the table. So this is only for things about getting what the next talks are. You can also just Google Grit City Think and Drink or UW Tacoma Grit City Think and Drink, and you can find not only what's coming down, but you can also look at recordings for the past talks that have been there. If you missed something, you want to check it out. We have a YouTube, all our stuff is up on a YouTube channel. The recording quality depends on whether Jim remembered to turn things on properly or not. So some of them are a little spliced together thanks to our wonderful audio people. So just so you know. Yeah, and that's my email. If you have any ideas, definitely email me at jimgeld@uw.edu and I will get a hold of folks you got interest in. So we do have the last day of giving away hats. We have been giving, we changed up our swag. So we have, I forget I'm wearing it. So we have shirts, hats, and socks. And now we're on this. These are the last. We're going to give away 3 tonight. Normally it's 2 a night, but we have 3 to give away. And then you need to come in December because the brand new bandanas will be out at that point. They do become collector's items because they are rare and hard to find, and we don't have that many. So make sure that you come and try to win again. Winning is free, so we give you a ticket. We will get those to folks who came in later, but then we'll give these away at the break before we ask questions. Okay. All right. I should thank, because I know I'll get in trouble if I don't, the sponsors of the program. So my bosses, the Interdisciplinary Arts and Sciences School at UW Tacoma. And I'm in the I'm in the Sciences and Mathematics department in that school now. And then the Office of Community Partnerships as well. So thanks to all of them for what they pay. They pay for the swag and they pay for the speakers. So it's a nice. So without further ado, I'm going to introduce our speaker tonight. Dr. Dan Holmes is a postdoc. Sorry, this is wrong now. Is a research scientist in the Fowler Lab at UW, where he works on developing tools to understand gene function. He got his PhD in microbiology from UW, working on a cancer-causing virus. That's a slip. And has previously studied prions in yeast and viruses in phytoplankton. He enjoys spending time outdoors with his wife, daughter, and their dog Trudy, 2 of whom are here tonight. So without further ado, let's welcome our speaker.
Okay. Hello. Let's see, can I stand and thing? Okay, I think that's good. Okay, I'm Dan. In my mouth. Okay. Okay, I'm Dan. For those of you who don't know me, my wife is a professor at UW-T. That's Leanne. Which is why I'm here. But I am a staff scientist in Doug's lab in genome sciences. And today I'm going to give a talk about kind of the background of the Human Genome Project, and then basically the data problems it created and how a lot of people now are working on trying and solve that. I'm going to do it through a little later in the talk through the lens of pathogenic mutations and clinical genetics. Go. No. If I have to, I can just.
Yeah.
Okay.
Those questions if they have.
Yeah. I'm just gonna go. Okay. Okay, so just a brief overview. We'll go over the Human Genome Project, and then here I put on NHGRI, which is the National Human Genome Research Institute. They actually have a whole page dedicated to a movement from the late 80s and early 90s of famous scientists that were actually anti-genome project. And they highlight quotes, which I copied and pasted some of them here, like the Human Genome Project, mediocre science, terrible science policy. But they also include copies of the full letterhead of the letters that they were sending to the NIH directors with their names and universities addresses attached, which I thought was pretty savage. So why did we need it? Which was a fair question. At the time, people did question it. So we'll talk a little bit about the science behind the Genome Project and the results and its shortcomings. And then we'll talk about the problem now, where we created this sequence, and now we've sequenced many, many, many more people. But how do we make sense of all that information? So we'll talk about different types of variation that we found, and then basically how we classically identified what mutants we're doing, variants we're doing, and then how now we're having to scale up and find new ways of doing that because the amount of data we have is too much for us to just rely on population genetics. So there's some background. People who don't know, the human genome is a lot of information. So 3.2 billion bases in just the haploid genome. So we have 2 copies of that because you have 2 of each of your chromosomes. So everyone in this room started with a single copy, including, for the sake of this talk, all the information that their body needed to make a whole person. This amounts to roughly 2 million pages printed as a book, and it has roughly 20,000 genes in it, or protein coding words, and organized into 23 pairs of chromosomes. So most of the goals of the Human Genome Project are actually kind of make sense. So they're trying to make a reference sequence of humans as well as a bunch of different models. They wanted to facilitate the development of new technologies. So they had the idea this would be like the space race, where it made a whole bunch of new technologies that changed life in many other ways other than just space exploration. And they wanted to foster collaboration across institutes and countries. And one thing that I thought was interesting that they thought to include at the time was that they wanted to understand the legal, ethical, and social implications of the work they were doing. So it's something that is now actually becoming a bit more of a problem. Like if 23andMe has folded and their genomic data became basically for sale, and it kind of creates a situation where we don't know what is going to happen with that information. So what made this project possible? And I will do a little bit of soapboxing here, basically because it was basic science that made all of this possible. So doing a lot of things that sometimes politicians on one particular side of the aisle like to think is not worth studying. So nematode genetics, so mapping nematode genomes as well as Drosophila genomes. So it's a microscopic image of a nematode, and then this is Drosophila melanogaster at the Mendel Museum in Brno, Czechia, with my daughter checking them out. Interesting. And then on top of that, E. coli and Saccharomyces. So the microorganism that is in all of our guts, as well as the fungi that made all the beer everyone's drinking. Understanding how their chromosomes were maintained and replicated was essential for the Genome Project, which we'll talk about in a minute. So we had to basically, it was too hard at the time, and even now we're actually getting better at it, but it's too hard to just sequence the genome itself. So we had to break it up into many different small pieces and then put them into bacterial artificial or yeast artificial chromosomes. And that let us create a clone. So organisms, including us, are much better at maintaining DNA than scientists are in a tube. So storing it in E. coli is a much better way to keep a stable copy of a part of the genome. And then you can ship it to your collaborators and you can work together on sequencing it. Um, so for the Human Genome Project, there were 33,000 E. coli clones that they had to use, and each one had about 200,000 base pairs. And then in addition to that, there was linkage mapping that was developed. So kind of classically mapping out the structure of a genome by, at first it was actual genetic characteristics like fruit flies. But later on, you could use radioactive probes to label a segment, cut it up, and then look at where it goes on a gel that you separate by size. And one of the architects of the Genome Project, Francis Collins, compared this to basically a roadmap where you're trying to go from California to Florida, but you don't actually know the direct route. But in order to map it out, you could, if you know a few spots, so say you know that the Alamo is in San Antonio, and say that the border crossing is in El Paso or something, you know where those two are. If you chop it up into random pieces, and then look at pieces and see how often those two things are segregate together, then you know they're close together. And then you can do the same thing for other landmarks and end up creating a linear map where you know what is close to what relative to one another. And you can use that information to scaffold deeper sequencing efforts. So it came together using these restriction fragment length polymorphism maps, is what they're called, that they can use to make a heritable element map of each chromosome. And they can kind of telegraph that onto a hypothetical physical map. And then they can identify which genetic element is in each of those E. coli clones that they have. And then they can go in and they'll use Sanger sequencing, which Leanne, maybe take out the slide that tried to describe that. But basically, it was the main technique used for sequencing at the time that let you do 500 to 700 base pairs at a time. So they would go through and do 500 base pairs, 500 to 700 base pairs at a time across each 200 kilobase clone. And then fairly often it would be a PhD student's entire dissertation was sequencing a clone. They would publish that and it would go into the project effort. And then they can use the genetic map to scaffold assembly of the entire thing. So what did this end up getting us? Basically, it ended up getting 92% of the entire genome. We'll talk why there was 8% missing at the time. It took us another 20 years to catch up on that. But it was also made up of about 20 people. So 19 individuals that were volunteers in the Buffalo, New York area. And then one individual made up 70% of the reference. Does anyone know who that individual was? Oh, no. It's someone that was involved in the project that famously spiked his own genome into it and got in kind of a lot of trouble for it. He was not known for being very nice in general, and he died last week. Yep, it was James Watson was the answer to that question. So yeah, he referred to himself as individual blended ancestry. I don't know if you've seen a picture of James Watson, but he looks like a white guy to me. But anyway, so what did all this cost us? Actually, not all that much compared to just about any other. So $2.7 billion over 13 years, which sounds like a lot of money until you put it into context. So it's roughly a tenth of spending power in terms of what the space race cost us, and 1,000-fold less than the Iraq War, or whatever that's worth. And as of 2013, the return on investment of the Genome Project has been estimated to be about $113, $114 per dollar spent, which is pretty standard for NIH money. And just another soapbox, NIH money has been relatively flat for about 25 years. So there's been some increases in funding, but it has not kept up with inflation. So it's not a big difference maker here, but if you have family in Indiana that you can go and tell them to call their Congress people or senators, that's worth doing. So what is this draft genome missing? Most of the sequences that are missing are repeat rich. And the reason why I think is relatively kind of intuitive. But basically, if you have a lot of repeat rich sequences and you try to align them, in this case, it's showing different elements. So 2x duplicates here, 2y's and 2z's, showing you that as the similarity between the two, so these are identical These are a little less identical, and this is only about 70% identical. Your ability to actually distinguish identical things if there are duplicates is non-existent when you have very short reads. So it wasn't until 20 years later when we had technologies that let us sequence many kilobases at once that we could actually resolve all of this. And famously, people joked that once we could, we realized there wasn't all that much of importance in those regions of the genome. So there aren't a lot of genes here. It's a lot of regulatory elements that are doing things. So the non-coding people always get mad when I say things like that. But I think the main thing that I think is important from all of this is that there isn't just one human genome. There are many. So this was taken at the time that the full genome was actually sequenced. This picture was from a News and Views article. But it's just giving examples of how if you have your reference sequences, you can have individuals that you go back and sequence and they have duplicate sections, areas where they have double of a certain sequence of DNA, where they've had a duplication event occur, or some that have had deletions, and then some that have sequences that we haven't seen in a reference collection yet. So just how many human genomes are there? Like, do we want to get an idea of that? Oh, and the Genome Sciences Department recently, another lab has gone through and actually gone out and asked basically for mother-child or parent-child pairs, like how much similarity is there between the parent and the child? And they refer to it as transmission events, which seems kind of impersonal to me, but basically in each child, there are 98 to 206 de novo mutations in the child that aren't present in either parent. So it's just by chance these changes are accumulating over time. And this is only germline mutations, mutations that the child is actually inheriting from the germline of the parent. So if you look at somatic mutations, it gets much more messy. So germline mutations are things that are in your parent's germline, so they end up getting put into the fertilized egg, and then they become a person. Somatic mutations are not present at fertilization, but they are acquired somewhere either very early or any time in the course of your life in one of your cells. So they end up making appearances in the adult, but then they, except for very rare cases, are not inherited in the germline. So how many somatic mutations are in each and every one of us? And people have actually asked this quite a bit across many different species, but in humans, there's actually a lot of diversity in individual people. So this plot is showing mutations that accumulate over time, and this is the x-axis here is age in years. So as we get older, we accumulate more and more. And it's important to note that it's really hard to know exactly how many are in a person. This is taken from blood, where they just take blood and then ask what kind of variation is showing up in the person's blood. So you can't take a whole person and take all their cells, and it's destructive sampling, so it wouldn't quite work out. But where are these coming from? Environmental factors probably play a role. So tobacco smoking leads to increases in somatic mutation. Drinking alcohol actually leads to increases in somatic mutation. But most of it is actually stochastic, which is actually stochastic, which just means it occurs randomly as DNA is replicated. So I mentioned earlier that E. coli and yeast are much better at maintaining DNA than we are in tubes, but in reality, it still makes mistakes. You go over enough bases. And as an example of that, so just what is the consequence of this is cancer. So this is from a kind of I guess now it's an older paper, but it's like 10 years ago. But it's comparing divisions from stem cell and incidence of cancer in that particular cell type. So I'll clarify a little bit. Basically, all the cells in your body have a precursor cell, it's a stem or stem-like cell. And then in order to get to their differentiated form, the differentiated form is like the end state that's usually quiescent, which means non-dividing or nearly non-dividing, where it does its job. There's a different number of divisions for different cell types. Some of them just need a few to get to their terminal form, which are the ones down here at the bottom. But some of them, particularly like colon cells, the colon epithelium, are kind of farther at the end, which means they take many definitions, which means many more replication steps, many more times for DNA polymerase to make a mistake. So this actually works out to a pretty clear linear correlation between cancer incidence, which is on the y-axis, and the number of divisions that have to occur. One of the conclusions from this paper was just that most cancer, they estimated somewhere around 2/3, is actually just a result of stem cell, like cell division, which I actually thought was kind of liberating. So it's, I don't know, a lot of times in disease, it's people tend, I don't know if it's just our society or because I was raised Catholic, but people tend to like assume that there was like blame on the patient fairly often. But most of the time, it's not. No one's fault. It's just something that happens, and we have to deal with it. Um, so what do these mutations look like? What is actually happening at the molecular level? Kind of go through an order of severity. Structural variants are one thing that happens. These actually happen all the time, but most people don't notice. So these are large deletions in chromosomes, duplications, inversions, so sections flipping around, or swapping between chromosomes. And this is actually the leading cause of miscarriage, which is also something that actually happens very common. It is very common. Miscarriage happens because these cases are so severe that they're non-viable. But you can also have much smaller things happening. So at single nucleotide level, you can have insertions, deletions, or repeat expansions or contractions, and the nucleotide variants, which are going to be the most talk just of our work. Um, and then so a few other flavors to talk about real quick are coding versus non-coding genetic variation. So in your genome, you have genes and then other sequences around it, which some cases play regulatory function, some cases they're just hanging out. So non-coding are in red, and then coding is in blue. Our lab focuses almost exclusively on coding changes, like things that actually change the peptide sequence. So if we want to zoom in on that, if you have a sequence here, so it spells think and drink, is your wild type sequence, you can have a synonymous mutation. Let's say it changes this C to a G. So now you still have think and drink. It doesn't change the actual encoding peptide. If you change this C to an E, it's now think and drink, which doesn't quite mean the same thing. And then you can have frame shift mutations. So a single deletion now causes the reading frame of the ribosome to actually be out of frame. So now you end up with a bunch of nonsense getting translated. So this one's obviously going to be more severe for an important protein than one of the others. Whereas this one, typically, there are a few people that get angry when I or my boss says this, but synonymous mutations typically don't do anything. So we use them in our work as a control, basically. But missense mutations are frequently unclear. So this could do nothing. Alanine to aspartate is actually a significant change, but it may not matter for the protein where it's actually occurring. So this would be a case where you actually have to go figure out what's going on. So how can we tell a benign mutation from a pathogenic one? If we have a patient that comes in that I'm not a clinical geneticist, but I work with a lot of them. So I'm not a clinical geneticist, but I work with a lot of them. So I always have to go and talk to them about this sort of thing. But they frequently will have someone come in and they're presenting with cancer predisposition in their family, and they have their genome sequenced and they have a mutation and they want to find out what's going on. So how do you go in and figure that out? Classically, this was done by pedigree studies. So you would go and collect information from all their family members. So you need to have enough people available to actually be able to demonstrate causality. So it needs to be a relatively common disease, a relatively common allele, enough that you can actually figure this out. And this is actually considered a seminal paper now by Mary Claire King from 1990. So this is BRCA1 pedigree. And actually at the time, they didn't know the name of the gene. They just had the part of the chromosome that they had identified. In addition to that, polygenic and multifactorial diseases are actually much harder to figure out. So if there are many different gene mutations or variants that are needed for a disease to actually happen, it's going to be hard to figure that out in a pedigree. If there are environmental factors involved, its penetrance may make it so that pedigrees are not that useful. So can we just solve the problem with more sequencing is one thing that we are actively trying. So sequencing has been increasing at a drastic rate, so much so that we can't, like the image from the flyer was the amount of data needed to store everything is kind of drastic these days. But But basically, this map is showing in this is gray the number of variants of uncertain significance. So things that we see in humans in sequencing data that we don't know what they're doing. This is taken from ClinVar, which is one of the main repositories that clinicians can go and deposit variants when they have information on them. So it's coupled with phenotypic data, so it can be used to actually draw conclusions. So most pathogenic variants actually tend to be fairly rare, and there are some benign and likely benign. And actually, in 2019, they changed the criteria for how to define likely benign, so there was a jump up in those. But the number of VUS, variants of uncertain significance, that are accumulating is kind of outpacing our ability to figure out what they're doing. And one question I think is important to ask is it a problem that almost all of our sequencing is of Europeans? And that's true. So here is individuals in GWAS studies, which are predominantly of European ancestry, which is pink here. And there are active efforts to try to ameliorate this, some of which are in Seattle in our department. One of them is this NIH initiative called All of Us, which the goal of All of Us was to create like background equitable genome sources for doing exactly this sort of thing. So inferring what different variants are doing. We'll say most of the NIH administrators that were responsible for this have been fired, and the funding has been curtailed. So if you have family members in the middle of the country, you can please call them. But if the initiative is pretty interesting, but part of it was actively working with tribal communities to get information from them, and then to work with them so that they still maintain ownership of their data. But one thing I like to stress is that this wasn't just an ideological effort. So you can have population bottlenecks where a variant that's rare in the European population could actually be fairly common in some subgroup somewhere else in the world. So that gives you kind of a natural experiment to ask what that variant's doing. And you can have many different bottlenecks like that around. So if you're sequencing deeply, you can kind of, one group helps another. It's more of a rising tide raises all ships situation rather than an ideologically driven decision. So are there any other ways to identify pathogenic variants? I'm actually finally going to get it. I'm actually an experimentalist, not a population geneticist. This is the busiest slide I have, and this was the best example of an assay that we run. So this is called saturation genome editing. So this is a way of using laboratory techniques in a wet lab to try to reclassify genetic variants. And basically, it's taking a cell line where, and I'm still sticking with BRCA1 just for consistency. So there are a bunch of variants in BRCA1, even though it is a highly penetrant disease that just aren't common enough for causality. So we make a library to edit the locus of a cell line that is, it's called hap1 because it's haploid. So it only has one copy of the human genome because it's someone found it by chance and it happened to be messed up enough to be useful. But BRCA1 is essential. So if you knock in mutants of BRCA1 and those proteins are now non-functional, the cells will die off. So you can tile across portions of the genome that you know are important. So they chose two different sections, the ring domain and the BRCT domain. And knocked in a library of variants. So you have this pool of cells where each one has a single variant knocked in. And then you allow those cells to grow for 3 weeks. And then at the end of it, you sequence the locus. And then you can identify, in this case, they're showing single nucleotide variant 4 and single nucleotide variant 1, which are losing functionality, which means they're depleting over time. So then you can, this initial paper was just showing that you could do this. They weren't even going back and trying to reevaluate clinical variants. And then a few years later, we had another paper where we were trying to answer this question. So you can find variants that harm cells in culture, but what does that actually mean for patients? And they were taking this data and data from 2 other tumor suppressor related screens that we had done, and then asking, how does this compare to known clinical variants? And then how can you use that to recalibrate our current scoring of variants of uncertain significance? And the answer about how it does against known variants is actually pretty well. So this is the function score taken from the saturation genome editing assays. And this is just looking at known variants from ClinVar. So in blue, we have benign variants, and in red, we have known pathogenic variants that are known to cause cancer in a patient. So in known variants, it does very well. So this gives a fairly high confidence for its ability to score variants of uncertain significance. So they use something called a naive Bayesian classifier, which I do not entirely understand what that means. But they're able to use it to reclassify variants of uncertain significance for BRCA1. And basically, they're able to move 54 of the 110 variants of uncertain significance in ClinVar over to either benign or pathogenic, predominantly benign. And at the time this came out in 2021, it was actually relatively controversial. But since then, the American College of Medical Genetics is actually now accepting these kinds of assays as evidence for predictive function of genes in the clinic. And our group actually supports MaveDB, which is a database of all the different assays that we've done and other groups have done, that we try to use fair data principles to collect it so that people can do other things with it down the line. So if someone wants to, it's there and you can download data from millions of variants. And then just a few weeks ago, we have MaveMD, which anyone can access, but it's basically designed for clinicians to go, if they get a report back that a parent has a variant, they can go and look at the actual data and see what it's telling them across many different modalities. Although the clinicians I work with want me to stress that you should not try to diagnose yourself. It's very easy to get your own genome sequence if you have $1,000 sitting around, which is a lot, but you can do that now and see what you have, and you'll find some things, but you really need the help of an expert to actually inform any decision making. So what kind of variants do these things resolve? One of the interesting things that we've published is that it's if you look across communities, these assays tend to selectively resolve variants in communities that are traditionally underserved. And the reason is actually, I think, fairly obvious, and that's just that these are background agnostic. So in our case, we're not selecting, when you're doing genome sequencing and you're picking a certain population, you're skewing what you're actually going to see. But in our case, while we do a lot of clinical variant work, because that in part helps pay the bills, most of us are actually more traditional biologists that are kind of interested in figuring out what the proteins are doing. So when we design our assays, we try to make every single possible variant at every position. So we're making 20, usually 22 changes at each site. But with MAVES, this is taking this BRCA1, P10, and tumor suppressor P53, and all the VUS that are in ClinVar for those genes. So in blue, it's non-European background, and in orange, it's European backgrounds. And so there's a few hundred more, 150 more VUS in the non-European background in ClinVar. But then if you use MAVES in the same way that we just described, using a classifier based on functional data, the number of VUS ends up dropping to basically a statistically indifferent number between the two populations. And this was done by an MSTB student in our group. That is there's a lot of statistics in it, some of which is over my head, but it is an interesting thing that I had not thought about. So how easy are these actually to do? And the answer is that it's not that complicated. So this is actually something that I've actually been heavily involved in. It's using computational tools and some cloning techniques to try to scale up our production of assays of variant effect. So we can basically use oligo pools instead of traditional synthesis techniques. So the main company we use is Twist. Yeah. You can order whole libraries from them, but they cost maybe about $40,000 to $50,000 for a 500 to 400 amino acid gene, which we've done in the past. That takes up a lot of grant money. So we switched to doing this, where we order a pooled format, and then we use a computationally designed set of primers to retrieve individual piles of genes from them. and then synthesize wild type gene blocks to go with each tile, and then clone them using an assembly type called Golden Gate, which is actually just very efficient, but the enzymes for it are very cheap. And I guess the endpoint is that lets us basically make 20 or so libraries of these a month for about $3 per position, which has been scaling up our efforts. And then just I'll talk briefly on what we're doing with this. We're basically been targeting using our maze to target rare disease. And a lot of this has been done with funding from the Chan Zuckerberg initiative, which is a little weird, but it has, also, yeah, like, yeah, everyone has their benefactor, I guess. But Priscilla Chan is a pediatrician by training. So a lot of her friends work with rare patient groups. So that's where her interest in this comes. So they have a collection. It's called the Rare as One initiative, where they're trying to help connect patient groups to scientists that can help them with their disease of interest. So we are currently completing a pathway scan of 22 members of the MAPK pathway, which is very important for somatic cancer. But it also causes a developmental disorder called Noonan syndrome. And then on top of that, we also have currently working on creatine deficiency disorders and RIN, which are both developmental disorders where there are patient groups of parents with kids that want help. So I guess the lab has gotten fairly big, which creates some problems for me as I've transitioned to having more of a leadership role rather than actually getting to use my hands, which is more of what I wanted. But we have most of our funding is still coming from the NIH. This is from a NHGRI effort, and this is from a second NHGRI effort. And we've kind of been supplementing it with Chan Zuckerberg money and being. I guess that's all I have. This is just a picture of Rosie at the Pacific Science Center, and behind her is a quote from Rosalind Franklin, who Rosie's actually named after, that science and everyday life cannot and should not be separated. Anyway.
We'll give Dan a short break and give away some prizes. So if you have not gotten a ticket yet, let me know. Raise your hand. All right. Ooh. So we're gonna give these away, 3 of them. So look at if you're like me, pull out your reading glasses. If not, if you have better eyesight, you can see your numbers at the bottom. All right. The first hat goes to last 3 digits, 556. 556. Hey, there's a winner. They're soft. I can throw them. All right. So next winner. That's crazy. So the next 3, 557 is the next one. Hey. And more. Sorry. Random as possible. Okay. I know. Damn. 565. 5, 6. Hey. Now you can only find them on eBay. From those people who have them are going to be putting out for sale, so maybe you can get one there. But remember, in December we'll be back. The second Tuesday in December, we will be giving away brand new bandanas. At least I am hoping they're coming in by December. So brand new bandanas, and we'll be giving those away for a while. So we'll hope to see you again. But before we, so remember, next talk, we're going to bring up our speaker to ask quick Q&A. And thank you all very much. Dan, you ready? All right.
Junk DNA is a kind of a loaded term. I spend a lot of time acting like non-coding variation doesn't exist. Just because there's enough to learn about coding variation. I I don't know what the actual fraction would be. I think you probably something like in cell culture the number is actually probably pretty high. Like it could be 30 or 40% you can get rid of, and the cells would probably, you could find a way to do it and the cells will be okay. But I'm worried that in an actual person it's probably much lower than that. Probably single digits. I mean, well, I don't know. It is kind of weird though. Evolution is clumsy. It's like it's easier to add things from an organism than to take it away. So you do end up with a lot of extra stuff. It's just there could still be a consequence for getting rid of it. Yeah. Yeah. So it was a lot. Oh yeah, it was the telomere to telomere initiative. Oh yeah, so it was what were the extra parts like that last 8% that they had a hard time sequencing? It's repeat rich elements that are really hard if you're trying to align things together, like I had mentioned. It took very long read sequencing, so the lab, it's actually the floor above us, Evan Eichler's lab. He I think he's nice. We fight with them over space a lot. We share a tissue culture room with them, which is the genome sciences department at UW, as an aside, was built during a time where everyone was just sequencing. So that was founded in 2001. Um, but now everyone has switched to doing cell-based assays, like I mentioned, but they didn't put tissue culture space in really in the group. So everyone's constantly fighting for that. But they use long read sequencing to map those complex parts. So you actually have to get very long chunks of the centromeres and the telomeres and the ends and centers of the chromosomes to actually be able to map them correctly. That, yeah. Okay. So how many genes do we not know what they do? And the answer is quite a few. I guess the answer is we know what the important ones are for the most part. And now thanks to, so now we have assays, which is what I did in grad school, called CRISPR screening. So rather than trying to edit a precise target, you can knock out, knock down, or increase expression of every gene in the, or everything that looks like a gene in the human genome, and then ask if it's important either for survival or for a particular function. So people have kind of gone through and scraped through the remaining things that look like they're essential in cell lines now. There's still a lot that we don't know. Um, what would a lot of stuff that looks like it's neurological or involved in sense. Like they'll get a label because it looks like an olfactory receptor, but there are tons of genes like that we have no idea what they're actually doing. In terms of actual percentage of the genome, I probably should know the answer to that, but I don't. Yes. Oh, fun and accurate. Was it most scientists like Gattaca? Uh, yeah, with a yeah, um, yeah, I don't know if Gattaca's fun, but unfortunately, it's probably sometimes science fiction can be um, prescient. I don't know. Yeah, I don't know how I don't maybe we'll pay that much attention to this, but there's kind of a movement towards like personalizing gene editing, which I don't particularly like. But you can find a lot of um, there's like one famous influencer that took a lentivirus, a modified HIV, and put it into his bloodstream that expressed green fluorescent protein, and then showed that he could see it in his cells. And I, yeah, you shouldn't do that. But there are companies now that are based around trying to select like edit embryos for desirable characteristics. You can find people in Silicon Valley are really into that idea. So there's a lot of money flowing into it. That um, I think so I guess I can talk about that. I'll stop talking about that. But Gattaca is an interesting movie. I don't know that there's a lot of bad portrayals of genome science. I think because it's fun. It the idea of being able to create something new that way is good for science fiction. But I'm sorry I didn't quite answer your question. Oh, well, yeah, there's a whole field that studies that now. So and there are people in my department now, Jay Shanduri, that actively studies that. But a lot of it, while there is this random element, a lot of it is like spatial patterning. So in a developing embryo, how would I don't know if I'm going to be good enough at answering this question. Basically, that there are sets of transcription factors that are receiving signals from how the embryo is organized. So you end up with patterning as the embryo's undergoing gastrulation. That will lead to development of the different layers, and then eventually development of all the different organ systems. Um, how would I? Yes, yes, but some things will become fixed. So like certain expression systems will become fixed epigenetically. So there'll be modifications to the DNA that suppressive chromatin. So some genes won't come back on, or some will be forced to stay on for longer periods. I think I had one point earlier in this talk where I said for the purposes of this talk, it had all the information needed, but that there are some other things now that exactly what's happening. So epigenome is upon the genome. There are modifications that are occurring on top of just the ATCG bases that are playing a role. But if you're really if you're really into that, I can point you to put places to look to find more information. I don't. It is an area that we're still trying to figure out. Some model organisms we know their developmental patterns pretty well, but mouse and human mammalian generally, we're still a little bit in the dark on how it works. So the question was, in addition to the experimental techniques, were there computational techniques needed to be improved? There definitely were. The alignment software that was used at the time, which has now since been improved upon since then. So it needed to be fast. Um, developing ways to store the genomic data was something that was also important, that most of the ways they were doing that then have carried over to now. They're still being used. I think probably the best answer for that is I don't entirely know, aside from I call the things at the command line now, but I don't actually understand how it works. But it is this was a large effort. There were a lot of software engineers that were then and still are involved in making it all work. So BLAST still gets used a lot. Um, when you're mapping to a genome, there's lots of other things now too, but bowtie still gets used a lot, although some people tell me I think the lab that made bowtie says we shouldn't use bowtie anymore, that there are better ways, but it's kind of ingrained, so everyone still does it. Um, has hammer is another very common aligner that people like to use. It's they all have their drawbacks. Some of them are better at finding non-specific, like kind of slightly different sequences. Some of them are better at making sure you have exact matches and doing it quickly. I guess does that answer your question? Are you interested in aligners? Is that those are the two common ones. But we predominantly use Python now. But a lot of it is actually even just calling at the command lines, using bash to run scripts over and over again on a directory of files, which tends to be fastest, mostly because I think what the things I'm calling are fairly often in languages I don't understand. So C++ or something that works a little better than faster than Python. Um, it is a one problem. One thing that our department tries to fix in training is making sure that there isn't too large of a gap between the software engineers and the biologists, but sometimes it still feels pretty substantial. Yep. So was there anything that they missed when they were planning out the genome project and they were considering the social, ethical, and legal implications? I guess one simple one is just sourcing where they were getting their genomes. Having a broader core, even of the first 20, I think would have, because the reference they ended up making influenced really even the work that's done now. It's still I still align to the within the lineage of the genome they created. I don't no one even though we have this telomere to telomere genome now, for some reason it still doesn't get used all that often. Um, I, I think I don't think they really anticipated the private sector getting so heavily involved. And that was even a problem from, I don't know if many people are, I didn't talk about Craig Venter at all, but there was an element in the Genome Project where they had kind of an insurgency from the private sector where venture capital came in and was trying to compete with the public effort. And that is kind of continued now with private sector companies having these collections of not just genomes, but also variant effects. So there's a few different companies now. Some of them are having a hard time profiting off of what they make, which kind of makes me happy. But that they have collections of patient information that they're sitting on with sequencing data, so that they could inform some of these clinical decisions, but they aren't releasing that. So I don't think. I mean, I guess it makes sense in some ways. These were all bureaucrats. They didn't think that was going to be a problem, but I don't know, and I don't know how they would have fixed that. But I'm sure some of them would say that if you could get them in a room where they um, wouldn't be recorded.
All right. Thank you all for coming. Hopefully, we will see you in December. I think it's December 9th, if somebody wants to check. Yep. December 9th is our next talk to talk about the sense of place in undergraduate education from our engineering faculty. And again, let me know if you have any ideas for other speakers in 2026. And if you wouldn't mind, when you take off, if you can put your dirty dishes and stuff in the bin back there and toss any stuff away, that will save the staff here a lot of time. Thank you all very much.